Hello and welcome to this webinar with the title Oral Biofilm, Friend or Foe. My name is Maria Payne and I am a researcher at NIOM within the field of microbiology. Before we dive into today's topic, I want to briefly go through some definitions for terms that I will be using in this webinar. So when we talk about the microbiota, it refers to all the microbes, so that includes bacteria, fungi and viruses in a particular environment. The microbiome, on the other hand, is the combined genetic material of all microbes in a particular environment. So for example, all the microbes on the tongue would be called the tongue microbiota, while the genetic material of those microbes would refer to the tongue microbiome. I will also be talking about both commensal and pathogenic bacteria, where a pathogen is a microbe that can cause disease, and a commensal is defined as a microbe that is not causing any harm. So what exactly is a biofilm? A biofilm is a structured community of microbes embedded in a slimy matrix that is attached to a surface. This slimy matrix consists of exopolysaccharides, proteins, lipids, and extracellular DNA produced by the microbes themselves. The ability to form a biofilm is almost universal amongst bacteria, and bacteria can form biofilm on most surfaces. The presence of a biofilm can give the microbes several advantages, and life within the biofilm is the preferred lifestyle for most. The biofilm helps protect the microbes against antimicrobial compounds from both the host itself or in the form of external factors like antibiotics and antiseptic compounds. The biofilm can also facilitate better communication and cooperation between the microbes, and the access to nutrients may also be enhanced. Of the biofilm present in and on our body, the biofilm forming on our teeth, dental plaque, is probably the best known. Let's have a look at that process. The biofilm formation begins pretty much as soon as the bacteria comes into contact and adhere to the tooth surface. This surface is covered by a thin film called the acquired enamel pellicle, which consists predominantly of proteins and enzymes from the saliva. Some bacteria, especially commensal streptococci, have specific surface protein enabling them to efficiently bind to the pellicle. These first adhered bacteria give rise to new binding sites where other microbes now can bind, either directly to the first bacteria or to substances in the newly formed biofilm. We refer to these initial microbes as early colonizers. As the biofilm matures, it allows for different microbes to colonize. Precisely how an oral biofilm is built and structured, what it consists of, and which microbes are present depends on several different factors, and we will have a brief look at some of them now. The microbiome changes as we get older. For example, following teeth eruption or other age-related changes in hormones and in the immune system. Also, differences among hosts like genetic factors or other health conditions and disease states will influence the microbial composition. Medications can play a huge role, especially those with the side effects of a dry mouth. The environment we live in will also influence both our habits and our diets, which again will influence the microbial composition and the biofilm. Within the mouth, there are several different habitats, and these habitats are important determinants for which type of biofilm that will form at this location. Oxygen levels, pH, and nutrients, for example, will differ between the different habitats. The mucous membranes in the oral cavities undergoes shedding, which prevents the accumulation of biofilm, while the dental surfaces do not. The biofilm formed here will mature and accumulate over time, which will lead to a change in the habitat. Other factors influencing the different habitats also include saliva production, clavicular fluid, and of course, oral hygiene. The maturation of oral biofilms is influenced by diet, oral hygiene, and the potential of shedding. Newly formed biofilms are very different to a more mature biofilm. This is mainly due to the changes in density, making the interior of the biofilm gradually more anaerobic as the biofilm thickens. The changes in density will also impact and alter the microbial interactions and the immune responses triggered by the host. Now that we have an overview of the many factors influencing the formation and the type of biofilm, let's look at some examples of oral biofilm. In this first example, we will look at biofilm formation on the tooth surface. First, the early colonizers, which is predominantly commensal streptococci, adhere to the pellicle-covered tooth. This creates new and additional binding sites, which allows for other microbes to bind, and the biofilm production is initiated. The main component in dental biofilm is EPS. EPS is short for exopolysaccharide, which are long chains of carbohydrates. A few of the typically early colonizing bacteria are shown here, and these actually perform several important tasks that support a balanced microbial community within the biofilm. Some commensal bacterial species contribute to a healthy balance by maintaining or increasing the pH, while other bacteria can produce compounds that either kills or prevents growth of the more pathogenic bacteria. Both the increased pH and the toxins produced by these bacteria inhibits the growth of the more notorious Streptococcus mutans. Mutants, on the other hand, can counteract the commensal bacteria attack. As long as sucrose is available, mutants can produce lactate, which reduces the pH in the biofilm. Most commensal bacteria will not be able to survive at an acidic pH. Another important Streptococcus mutans trait is its ability to produce exoenzymes. One exoenzyme, called glycosyltransferase, is an enzyme that can produce glucans, which are very important building blocks in the cariogenic biofilm. The production of glycosyltransferases increases when the pH decreases and when the environment is acidic. The glycosyltransferase enzymes can bind directly to the pellicle, but it can also bind to other microbes, and thereby converting them into glucan producers as well. This contributes significantly to the biofilm biomass. With high production of EPS, the local environment within the biofilm gets more acidic, and this is partly due to EPS trapping the acid produced inside of the biofilm. As the environments get more acidic, it promotes the growth of specific bacteria that is able to tolerate an acidic environment. Here are some examples of typically promoted microbes in this type of maturing biofilm. These are all acid-tolerant microbes which both collaborate and promote one another. A sugar-rich diet will stimulate the collaboration between these microbes, which will maintain the increased production of both GTF enzymes and acids, and thereby lowering the pH even further. As the pH decreases, the diversity in the biofilm is also reduced. The microbes present in the biofilm are acid tolerant, and they contribute to maintaining the acidic habitat by producing more glucans and more acids. If the pH in the biofilm continues to decrease, it will eventually lead to the enamel demineralization, resulting in the start of or the progression of dental caries. Mutants often play an important role in the development of cariogenic biofilm, but it might well be absent at the time dental caries occurs. Acid-tolerant and acid-producing bacteria are the main cause of dental caries, but there are several different species of bacteria that also fit this description. This biofilm could, and most likely would, lead to caries if it is not removed in time. Removing all sugar from the diet would prevent a cariogenic biofilm, but a biofilm would still develop and mature, but at a different pace and with a different structure and with different properties. Even though a sugar-deficient biofilm would not lead to caries, most, if not all biofilms, would still cause a problem if they were allowed to mature for long enough and without any disruptions. We will now have a look at a different type of biofilm. As the biofilm matures, different microcommunities or niches are formed within the biofilm. There will be localized gradients of oxygen and of pH. The biofilm formed in the gingival pocket will, as the biofilm matures, have little to no oxygen. In addition, the nutrition for the microbes in this habitat will mainly come from the gingival clavicular fluid and not from the saliva. The gingival clavicular fluid consists mainly of glycoproteins, but also the immune system's own antimicrobial factors. The bacteria establishing at this site is most often anaerobic. They need the absence of oxygen to grow, and they are proteolytic. They break down proteins, not sugar, for their energy. The breakdown of protein most often results in more alkaline products, which will increase the pH locally. Most of the bacteria found in the gingival pocket are also gram-negative. They have an outer cell membrane consisting of lipopolysaccharides, LPS, which is strongly immune stimulating and contribute to the local inflammation. Additionally, many of these bacteria also have properties which offers protection against immune mediators. If not removed, these microbes will cause inflammation and most likely lead to periodontal disease. As seen from these examples, the microbes that cause caries and gum disease are very different and with different metabolism and different preferences. Deciding whether a microbe is good or bad is totally dependent on the environment. We have now looked at two examples of harmful biofilms, where different microbial communities lead to different disease states. But a biofilm can also be good or beneficial. A beneficial biofilm has a good balance and composition of microbes that is tailored to that specific host. As seen on previous slides, there is a wide variety of both external and internal factors influencing the composition of a biofilm, and these factors determine what the healthy biofilm will look like for the specific host. Regardless, we can still give a generalized overview of which type of bacteria a good biofilm should consist of. So a good or a beneficial biofilm will contain microbes that can protect the host by counteracting pathogens, either by direct inhibition, for example by producing toxins, or by occupying the space by simply being better adapted to that specific environment. Some oral commensal bacteria have both pro- and anti-inflammatory activity, and this is crucial in order to maintain a healthy balance at the strongly colonized surfaces in the oral cavity. The immune system must be stimulated a little, but not too much. Balance is the key. We know that bacteria in our gut help us break down certain nutrients. But what about the mouth? Some oral bacteria can break down nitrate to nitrite, something our cells are not able to do. Nitrate is a compound found in several vegetables, like beetroot and leafy greens. Nitrite can be further reduced to nitric oxide, which has been shown to inhibit the growth of some cariogenic bacteria. So, good or beneficial microbes can aid us and help us deal with the bad guys. But Can we then remove too much of the biofilm? So, normal dental hygiene with brushing and flossing doesn't remove too much of the biofilm. But it removes enough, and it actually facilitates the good and early colonizing bacteria to quickly recolonize the sites. But using substances with a broad antimicrobial range, like chlorhexidine, appears to have a very negative effect. Recent studies have shown that the use of chlorhexidine-containing mouthwash reduces the bacterial diversity in the mouth. In addition, the amount of lactating glucose in saliva increases, while nitrite, pH, and buffer capacity goes down. The reason is that chlorhexidine reduces the number of bacteria able to break down nitrate. When bacteria reduce nitrate, they often use lactate as a carbon source, which prevents a drop in pH. Less nitrite leads to less nitric oxide, which in addition to having an antimicrobial effect, also is an important factor in the regulation of blood pressure. Daily intake of chlorhexidine in this study led to a small but significant increase in blood pressure. This is suggesting that beneficial bacteria in the mouth plays a rather important role, and not just locally. Oral bacteria can lead to caries and periodontitis, but can these bacteria also cause problems elsewhere in the body? Over 30 years ago, a Finnish cardiologist, Kimmo Mattila, noticed that most of the patients he had on the operating table for heart attacks had really bad teeth. Mattila and his colleagues later confirmed a significant association between heart attacks and poor periodontal health. This connection with cardiovascular health has since been extensively investigated. An association between gum infections and conditions such as cardiovascular disease and diabetes is particularly well established. Findings in these past few years have also linked periodontal infections to other conditions such as Alzheimer's disease and rheumatoid arthritis. Actually, today, periodontitis has been found to be associated with various systemic diseases. But decisively confirming these associations, however, has proved challenging. First, conditions such as cardiovascular disease and diabetes takes decades to develop. And secondly, to demonstrate causality decisively, these studies would have to deny a cohort of people periodontal care and see how it affects the incidence of disease. Such studies would be highly unethical, but observational shorter-term study can still give very good data. Knowing all this, that the bad bugs in the mouth can potentially cause trouble elsewhere in the body, is there anything else we can do besides limiting sugar and practice good oral hygiene? I now want to draw your attention towards probiotics and prebiotics. Most of you are probably familiar with probiotics for the gut. People often take this as part of or after a course of antibiotics to re-establish the gut microbiota. Probiotics are defined as beneficial microbes that, when administered in adequate amounts, confer benefits to host health, while prebiotics are defined as compounds in food that induce growth or activity of beneficial microbes. Good candidates for oral probiotics has been found. Streptococcus tentisani, for example, has proven efficient in killing certain pathogens, while species of Rothia are efficient in breaking down nitrate, which can also inhibit the growth of pathogens. The challenge with oral probiotics is both how to deliver them efficiently and also how to get the probiotic species to establish in the mouth. And this is something that researchers are still working on. Another approach to changing the oral microbial flora is to provide specialized nutrition that will stimulate a healthy balance and composition of bacteria. Toothpaste with arginine is already on the market in some parts of the world. some commensal streptococci can break down arginine, which increases the pH in the biofilm and thereby inhibit the growth of acid-loving bacteria. Another approach that has been suggested is adding nitrate to toothpaste, which can be used to selectively feed bacteria that promote good health. Before we end this video, I just want to give you a short summary of what I consider to be the most important points. The microbes in our mouth have important roles. We should not be aiming at eliminating them completely. We should, however, disrupt and thereby facilitate a good balance. Good oral health might be more important than we originally believed, as bad oral health is associated with a whole range of illnesses, which you likely can help preventing or at least slowing down by taking care of your mouth. The best way to do this is to have good oral hygiene and a healthy and a varied diet. And lastly, one should be careful with using anything with a broad antimicrobial effect, as this might lead to several problems later on. And that concludes this webinar. If you found this topic interesting and would like to learn more about the microbial life inside the mouth, we offer a tailor-made course on the oral microbiome. We also have other interesting and specialized courses on different topics which you can find on our webpage niom.no/courses. Thank you so much for watching.